Hi, everyone. Thank you all for coming to my thesis presentation. My name is Catherine Nickel, and I am a biology and Spanish major, and I do microbiology research with Dr. Edwards on yeast. I am currently presenting my research at a conference in Houston, so I was unable to be physically present at the Honors Symposium, but I have prepared a presentation via recording. So we're going to go ahead and get started. I will be presenting on the standardization of Saccharomyces cerevisiae microplate reader covering parameters, and more specifically, how covering parameters can affect evaporation and how evaporation can affect growth. In my presentation today, I have four main sections: the introduction, methods, results, and conclusions and future directions. We are going to start with the introduction and understanding why yeast is important to us and what we can use it for. So, what can yeast do for us? A few challenges that the modern world faces include global warming, reliance on fossil fuels, and food and energy security, all of which can be addressed with biofuels, or more specifically, second generation biofuels, which do not compete against food supplies because they are based on non food raw materials or non food sources. Essentially, we can combat these challenges by increasing the use of biofuels, more specifically second generation or higher. So you may be wondering how this relates to yeast. Well, yeast, or in this case, Saccharomyces cerevisiae, can convert biomass to ethanol via fermentation, and this ethanol can be used to make biofuel, specifically second generation biofuel. This biofuel can be used as an alternative to fossil fuels, and fossil fuels are finite resources that emit greenhouse gases when combusted and negatively affect the environment. So, replacing these with biofuels can help with all three challenges previously mentioned: global warming, food, energy, and water security, and reliance on fossil fuels. As a lab, we are working towards developing fermentative microorganisms with the properties for effective industrial production of bioethanol. But in order to do this, we need to determine the best conditions for microbial growth in order to increase fermentation to optimize the yield of bioethanol. And in this case, microbial growth is equivalent to the ability of the organism to ferment ethanol, or in other words, the better an organism is growing, the more likely it is to ferment more ethanol. So we're assuming that increased growth equates to a better ethanogen. Essentially, growth can be used as a proxy to measure ethanol production, but how are we going to measure growth? So there are many methods to analyze and assess microbial growth, but for this research, we chose to use the plate reader method because of its various advantages, but also because we wanted to improve its disadvantage. So let's talk about how the plate reader method works. A microplate reader is used to measure absorbance or optical density. So if you look at this simplified diagram here, you can see that the light strikes a diffraction grating, which separates the light into different wavelengths, and the grating is rotated so that only a specific wavelength of light interacts with the sample. The light absorbance by the yeast is measured as its absorbance or its optical density. And this is indicative of the density of the cells in the well. So when measured over time, we can track the cell growth or how the number of cells in the well increases over time. And there are some advantages and disadvantages to this method. The microplate reader does allow for a quantitative and high throughput analysis of bacterial growth or microbial growth for repeated experiments with results that are reproducible, which does save time, effort, and material by allowing for the incubation of up to 96 cultures at once. However, there is a lack of standardization of the microplate reader parameters, which can lead to a lack of reproducibility in research experiments. So we're going to discuss a few research articles that looked into this lack of reporting of experimental parameters and how that lack of reporting affects reproducibility. As I previously mentioned, microplate readers are great for experimental reproducibility, but without a field-wide standard parameter, the reproducibility of microplate reader experiments as a whole can be challenging. So here's a study conducted in 2016 that revealed that 68% of the 100 research articles included in the study did not report shaking parameters, and 76 did not report the covering method used. Additionally, the research articles that did report the shaking parameters and covering methods often used different parameters for both. So the plate reader itself allows for reproducible experiments, but the lack of a standard method and standard parameters for recording microbial growth does handicap the experiment's reproducibility within the research field. And this lack of reproducibility can in turn affect experimental results and conclusions. So before we dive into the research showing how reproducibility can affect experimental results and conclusions, it's important to know what we will be talking about. So here is a typical 96 well microplate. It has 96 wells that can be filled with low volume cultures. There are also plates with more or less wells, but the 96 well plate is common throughout research. These plates can have various different types of covers. For example, there could be no covering. You could use the standard plastic lid, which comes with the plate. You could use an adhesive sticker covering or a more specialized covering such as the microclimate environmental lid, which, as you can see here, has a piece of felt in it, and sterile solution, typically water, can be added to the felt to dampen it to increase humidity of the plate and of the wells, in order to decrease evaporation. Now that we have a better understanding of the research tools, we will look into the experiments themselves. So here are a few examples of how this lack of reproducibility can affect experimental results and conclusions. And the tendency not to report covering methods in journal articles is significant because studies have shown that different covering methods can affect the experimental outcomes. Here on the left-hand side, there was a study that looked at the effects of 3 different covering types on evaporation of media from the wells. So you can see that there is high evaporation from the periphery wells when using the standard plastic lid, and when using the oil covering or the sticker covering, there is decreased evaporation throughout the entirety of the plate. On the right side over here, you can see there was a similar study conducted looking at percent evaporation using different covering methods. And this top covering type, competitor A, shows a similar pattern to the standard plastic lid covering in that there is high evaporation from the outer wells, which is known as an edge effect. This bottom covering type shows decreased evaporation throughout the plate and no edge effect. These studies found that covering methods are directly associated with growth medium volume decrease via evaporation, which in turn can affect microbial growth. And this leads into my purpose of research. The goal of this research was to determine the covering type with the best evaporation inhibition, which can be used to standardize microplate reader protocols in order to increase reproducibility in future research on second-generation bioethanol production from yeast without negatively affecting cell growth. So to explore my research question by looking into the effects of covering type on evaporation and the effects of evaporation on growth, we're going to go through each procedure and their corresponding results. In order to calculate percent evaporation, I needed to get the average weight of the media I would be using. So I weighed 200 microliters of tryptic soy broth, or TSB, using an analytical balance. I repeated this 5 times to determine the average weight in grams of 200 microliters of TSB. And to avoid any confusion for anyone who doesn't work with microbiology or other fields of research, TSB is one of many types of growth media that can be used in microbiology research. So from here on out, I will be referring to it simply as growth media. So once I had the average weight of 200 microliters of growth media, I could start looking into the procedure for covering dependent evaporation. And 200 microliters of growth media was transferred into each well of a sterile 96 well plate. The plate was covered using the standard plastic lid, the microclimate environmental lid, a sticker covering, or not covered at all. The loaded plate with its covering type was transferred to a microplate reader and incubated for 24 hours under the conditions seen here. After incubation, the contents of each well were individually weighed in grams. This was repeated 3 times for each lid covering type, and the data was averaged and used to calculate percent evaporation. So I found that covering type does affect evaporation in plate reader derived assays. As you can see here, low evaporation is green and high evaporation is red. For no covering, there was high evaporation throughout the entirety of the plate. For the standard plastic lid, there was low evaporation in the inner cells or wells, but higher evaporation in the periphery wells. And for the adhesive sticker covering and the microclimate environmental lid, there was lower evaporation throughout the entirety of the plates, but the microclimate environmental lid did show this very slight edge effect. In addition to showing the percent evaporation for the entirety of the plate, I also separated the plates into well groupings. So you can see here, the outermost wells are blue and the innermost wells are pink. For the no lid, you can see there is increased evaporation throughout the entirety of the plate, whereas for the lid, sticker, and felt or microclimate environmental lid, there were decreased levels of evaporation. Here you can see that slight edge effect in the microclimate environmental lid. Overall, this shows that the adhesive sticker covering did not exhibit an edge effect and had the most uniform levels of evaporation throughout the plate. But what does that mean regarding growth? So we will now look into the connection between evaporation and growth. In order to look at the effects of evaporation on growth, S. cerevisiae was grown on TSA with 2% weight per volume glucose. A single colony of S. cerevisiae was used to inoculate 5 mils of growth media, which was incubated overnight for approximately 24 hours with the conditions seen here. The inoculum was then taken and its optical density at 600 nanometers was measured. This optical density was used to calculate the amount of inoculum needed, and once the correct amount of inoculum was known, it was added to 25 mils of growth media after agitated for approximately 1 minute. After agitation, 200 microliters of this media was transferred into each well of a sterile 96 well plate, and then the loaded plate with its lid was transferred to a microplate reader and incubated for 24 hours under the conditions seen here. This was repeated 3 times using the standard plastic lid and the adhesive sticker covering. Raw data from the growth procedure was exported from the Gen 5 software as an Excel file, and the data was used for 2 different types of analyses. The t-tests were run with Bonferroni corrections, and then data was converted to a CSV file and uploaded into RStudio to create a growth curve. For each experimental replicate, optical density was read every 15 minutes to generate a growth curve. This means that the growth curve is made up of a multitude of OD readings every 15 minutes, as seen in the figure here. Additionally, the minimum generation times and maximum OD 600 readings were noted. The minimum generation time is the least amount of time it takes for the yeast to double in number, which is important because it indicates the end of the lag phase and the beginning of the exponential phase, and it is essentially a marker that allows us to compare the growth curves of the 2 covering types. And the maximum optical density indicates the highest density of cells in the well, and therefore can indicate the most growth. And it's important to know the max OD to be able to compare growth between both covering types. By using the growth procedure and data analyses, we were able to generate a growth curve for both the adhesive sticker covering and the standard plastic lid. The adhesive sticker covering and the standard plastic lid had similar lag phases and final OD readings, but the adhesive sticker covering had a steeper slope, meaning the rate at which the OD increased was faster than that of the standard plastic lid. The average generation time for the standard plastic lid was 80.8 minutes, and the average generation time for the adhesive sticker covering was 75.6 minutes. And using a two-sample t-test, assuming equal variance with an alpha value of 0.05, it was determined that the difference between the generation time for the standard plastic lid and adhesive sticker covering was statistically significant. This indicates that the differences in evaporation using either covering type may affect the growth of the yeast, although other causes for this difference have not been eliminated. Additionally, the stationary maximum OD for the standard plastic lid was 0.02 AU, higher than that of the adhesive sticker covering, which again may indicate more growth. We also looked into the generation time and maximum OD at 24 hours by well location within covering types. The lines indicate statistically significant differences. As you can see for generation time, there are two statistically significant differences between the two covering types, seen here by the red lines. These differences are seen between the blue and yellow sections of the plate, which are the two outermost sections. This difference could be explained by the difference in evaporation due to the edge effect of the standard plastic lid. And within the lid itself, there was one statistically significant difference between the inner and outermost wells, the pink and the blue, which again could be explained by the edge effect. Within the sticker lid, there were statistically significant differences between all of the well groupings except for two. The innermost wells, green and pink, and the two middle wells, green and yellow. This tells us that there is greater variation in generation times for the adhesive sticker covering. And lastly, if you look at the maximum OD at 24 hours, you can see that there are no statistically significant differences between or within covering types. So, what do these results mean? What conclusions can we draw from them? And where can those conclusions lead us? So we concluded that plate covering affects evaporation rates in plate reader-derived assays. The standard plastic lids reduce evaporation rate compared to no covering, but they do result in an edge effect. The adhesive sticker covering had more consistent evaporation rates, no edge effect, and a shorter generation time overall, equating to overall higher growth rates, but there was more variability in generation time. And our overall conclusions are that the standard plastic lid is recommended for plate reader-derived growth curves, but the outermost wells should be filled with some type of sterile solution to avoid an edge effect and potential data skewing. Based on these conclusions, there are multiple outlets for future research. So we could analyze oxygen limitation when using the adhesive sticker lid covering and its impact on growth. The adhesive sticker lid covering had the lowest, most uniform levels of evaporation throughout the plate, but there were several statistically significant differences in generation time, which raises the question of the effects of other factors, such as oxygen limitation. And as discussed earlier, our lab's goal is to use this microplate method to study biofuel production from yeast. So future experiments could entail using this standardized microplate reader procedure to analyze effects of industrial stressors on ethanol-producing strains of yeast. Lastly, I would like to acknowledge a few people who have helped with this project, including Talita de Souza, Catherine Gonzalez, Cody Wegner, Bryce Jones, Jordan Macario, Derek Cardenas, Aaliyah Gutierrez Cano, Dr. Edwards, Dr. O'Keefe, and Dr. Stephenson. Thank you all for coming to my presentation today, and if anyone has any questions or comments, please feel free to email me at the email included on this slide seen here. Thank you very much.